So our next presenter is Dr. Jason Hinman from Sage Cerebro, and he'll be speaking about a multiplex immunoassay for vascular brain injury. Dr. Hinman is a physician scientist, stroke neurologist, and is an assistant professor of neurology at UCLA. He's the founder of Sage Cerebrovascular Diagnostics, and he completed his MD PhD at Boston University School of Medicine. He moved to Los Angeles and completed the adult neurology residency and a vascular neurology fellowship at UCLA. He then joined the faculty and set up his research laboratory in 2013. And as the founder of Sage Cerebro, he's focused on advancing new diagnostics for cerebrovascular health. Great. Join me in welcoming Dr. Jason Hinman.
Thanks, Casey, and thanks to Josh and the Luminex team for the invitation, the opportunity to speak today. A little bit different than some of the things we've heard today, but I'm excited to be here and tell you about the work we've been doing at Sage Cerebrovascular Diagnostics. What I'm going to talk about today is our efforts in the commercial space to develop a multiplexed immunoassay for vascular brain injury. And let me start with telling you why that might be important. So this is a patient of mine, he shown in the MRI scan there. He comes into the hospital with a small cerebrovascular event, showing the bright white signal on the left hemisphere of the brain, disabled from that stroke. It's, of course, a common and devastating neurologic disease, a leading cause of global disability and death. Diagnostic is particularly challenging because, while when patients come in, it's fairly straightforward. You get brain imaging and treat. But what we don't understand completely is who's at risk and how can we better modulate and understand that risk. So it's very unpredictable from that standpoint. However, it is associated with defined risk factors, some of which we can measure. We put the patient in the hospital. We might do cardiac echocardiogram to identify structural heart disease. We might do a cerebral angiogram or cerebral angiography to identify major vascular stenoses or lesions. We might put the patient on a cardiac rhythm monitor to pick up atrial fibrillation or another stroke risk factor. And we might do blood tests to measure common understood risk factors like cholesterol and blood sugar. What's lacking in the field is a recognition that the vast majority of the brain is highly vascularized by blood vessels that we can't measure. There's nearly 400 miles of brain blood vessels. We measure less than 3% when you come into the hospital and get an MRI scan. And when these blood vessels are damaged by the risk factors I mentioned, they manifest like this on the structural MRI with bright white signal, generally in the deeper areas of the brain. And this reflects a diagnosis of chronic cerebral small vessel disease. And what we're trying to do at Sage is develop a novel blood test to try to measure the damage that occurs to these small blood vessels, as it's a recognized risk factor for stroke and cognitive impairment over time. So thus then we started the company. We're currently based out of LA Biolabs in Los Angeles with the principle that we can take the signals that act on and produced by cerebral small vessels, intelligently cluster those, and then use that information to develop diagnostics for complex brain disease. And our first product is a blood test that will measure this brain vascular injury, and I think parallels the development of cholesterol testing for heart disease as a recognized risk factor that can be modulated. Our first in-class product, we'll be focusing on entering the dementia market, as chronic cerebrovascular disease is a leading risk factor for not only stroke, but also long-term cognitive impairment. And with that entrance, we think we can broaden access, accelerate diagnosis, improve the prediction and prevention of this disorder, and potentially reduce healthcare costs in the process. And importantly, the underlying principle is scalable and can offer innovative diagnostics for other neurologic diseases. Just briefly, this is my team. Most of the work I'm going to show you later on the slide deck is the work of our current lead scientist, Dr. Sadaf Pushi, who's been with us for the last year and a half, working hard on development of the assay. I'm going to start by showing you some of the published work that led to the intellectual property that we're working with at Sage. So in that work, we tried to identify the role of peripheral inflammation as a potential biomarker for white matter injury. We did that by studying 167 patients at risk for or having elements of cognitive disorder. They underwent a 3 Tesla MRI and a time-locked serum sample collection. And then we used a multiplex custom Luminex assay to measure several of the inflammatory molecules that are pictured in this network. Many of them had been previously associated with cerebral small vessel disease, but in this publication we made the link that these are part of a functional network of inflammatory signaling, and thus we're able to use them together to generate a single composite score that might represent the presence of MRI imaging, and thus advance a plasma or a serum-based diagnostic in adjunct with the MRI scan. What I'm showing you in the heat map here is each of those inflammatory cytokines clustered by the degree of white matter injury present on MRI. So going from left, low white matter injury, to high, you can see that there is a progressive increase in inflammatory cytokine signaling that can be associated with that white matter brain injury. That's shown again here in the scatter plots. This in panel B is the representation of the structural imaging that I showed very early on. We also used an advanced technique called a measurement of cerebral free water, which is thought to be a precursor, a measurement of cerebral edema that's a precursor of the structural damage that we're measuring here. And you can see that we also get a very strong correlation between our single composite score reflecting these cytokines and the measurement of free water. And representative MRI scans are shown on the right, separated by median values. So this represents the median in the high group and the median in the low group for both imaging techniques. Importantly, because we're interested in identifying the role of this signaling cascade in cognitive impairment, inflammatory signaling is quite hot in the amyloid and Alzheimer's field right now. In a subset of the patients, though not published in this article, we did do amyloid PET scanning and found no relationship between this particular cytokine cascade and the presence of brain amyloid, suggesting it's a unique vascular signal, which is what we're representing with these structural brain images. Then in a second related study, we captured patients at the UCLA emergency room who had come in with a complaint that might put them at risk for cerebrovascular disease. Our standard is to do an MRI scan, as I showed you earlier. In 131 subjects, we did the same multiplexed assay that I showed at the beginning on the Luminex platform. Here, it's more difficult to do the quantitative measurements of the MRI signal, so you can separate, based on the clinical scan, patients into mild, moderate, and severe. And I'm sorry, we're missing a representative MRI scan, which should be here. Not this plot, but what it does demonstrate, what the graph demonstrates, is that the inflammatory composite score as a function of the imaging represents a 94% specificity for the detection of severe silent brain injury. So with that information in hand, the University of California decided to patent that as a potential diagnostic for the detection of this condition. We're ongoing patent prosecution in a number of different localities around the world. The panel assay that has been exclusively licensed to Sage from the university is going to be tested in nearly 14,000 patients as part of NIH-funded studies ongoing in my academic laboratory, and has really been adopted as the fluid biomarker of choice for vascular cognitive impairment, leading to quite a bit of funding towards the lab. And in a recent SBIR grant that we put into to NIH, the assay was identified as it would clearly be used by the neurology and geriatric healthcare communities. So with that principle, then we try to move that to the commercial space. And so the simple workflow then is to collect a clinical sample, perform a multiplexed immunoassay, quantitate the measurement of the cytokines, reference them against our existing data, generating a single composite score that then can be associated with the degree or predicted degree of MRI brain vascular injury as detected by MRI. Importantly, that in order for this to be a commercially viable product, it has to be scalable, it has to be adaptable, and it has to be highly reliable with a fairly quick turnaround. So I was asked to talk about the challenges that we've been facing, the success and also the challenges we face as we've been working through this over the last year. And so what SOTF has been working on diligently is facing these challenges head on. So first, you first have to be able to identify your target, and that requires high potency antibodies. We've been fortunate to partner with Biotechny and utilize the robust antibodies that they have for the targets, the validated antibodies that they have for the targets we're interested in. Then we have to optimize that pairing such that we can improve the sensitivity. You, of course, have to minimize cross-reactivity. I asked Thomas about this earlier. And then two other challenges include optimizing the bead concentration and shifting the standards around since we're measuring multiple different cytokines that are present in serum and plasma at different concentrations. So I'll try to show you some data of what we've been working on all of these points. First, the Luminex XMAP technology is really optimal for the challenge we're trying to face because it allows us, obviously, to quickly scale and develop a multiplexed assay upon which we can add. So right now, our first product is a six analyte composite. We have IP protection on 2 or 3 other interested molecules and the ability then to scale up, although thinking about what Thomas said earlier about the upper limit of that scaling. Again, the audience is probably quite familiar with this. Another challenge is that we have to think about bead compatibility. So while we're developing the principal core assay using the FlexMap in the lab, our CLIA partner, with whom we plan to push this out as an LDT in the next year, is operating off the FDA-approved Luminex 200 device. So we have to consider that as we develop. And so then we decided to choose bead regions that would allow us to be maximally compatible with all XMAP instruments and provided enough distance between other regions that we could include future analytes. And so this is the principle that we use for our initial development. Again, the assay principle, this has been shown. Again, audience probably quite familiar by the time we get to the afternoon here, but essentially we have antibody-coupled beads that are specific for the various proteins that we're interested in measuring in the multiplex. We then add biotinylated detection antibodies that find those beads and then provide SAPE as a fluorescent indicator and then measure that out on one of 2 devices. Of course, the antibody chemistry, the coupling chemistry is, again, well known probably to this audience, but this is the technology, the chemistry that we've used. I know there are a couple different ones that can be applied, but we've focused on applying this chemistry here. We, of course, had to start by optimizing that approach. So here we first needed to test the relationship of the concentration of antibodies per million beads in order to optimize both that reaction and also our downstream analysis. And so you can see here in green is, I believe, 2.5 micrograms per million, and then 5 and 10. So the assay clearly saturates at 5 micrograms per million beads. Next, we have to measure the efficiency of that reaction. And so here I'm showing you the data across the multiplex with the efficiency of the coupling reactions as a function of the net MFI that we're measuring. And so we can skip through that quickly. Another core issue in a multiplex, particularly here, is the cross-reactivity between analytes. And so, of course, you want your bead-coupled antibody to find its target antigen, and then the detection antibody to find it again. And you don't want any cross-reactivity with non-target bead regions. And so what you can see in our 6-plex is that we have very high primary sensitivity with reasonably low cross-reactivity across the entire matrix here. We still have one analyte that we're working on a little bit, but mostly this is a very reliable assay with little cross-sensitivity. I mentioned the other challenge that we face, which is to optimize the measurement of the particular analytes that we're interested in, all at different ranges within the assay, or within the potential sample. And so here you can see a plot of the standard curves that we've generated in the initial development for the 6 analytes. In the shaded boxes are the target ranges, the reference ranges from our prior work. So you can see that we've tried to tweak that and optimize that so we're in the strong upslope of the curve for the majority of the analytes, and the concentrations not so relevant for the audience, but pictured here on the right. Again, detection across devices is a core issue as we develop this new diagnostic, and we're seeing some important differences in the sensitivity between the device we're using at Sage, the FlexMap 3D, and the Luminex 200, which is available at our CLIA partners. So we've had to do some re-optimization of the standard curves such that we can create the same kind of upslope within the target ranges on the Luminex 200. And so we've had to do some important work on that. You can see I don't know if the back of the room can read, but the net MFI detection is much more sensitive on the FlexMap 3D than on the detection device in the Luminex 200. So we're still working through some of this, but clearly there's some important differences at this early stage. Next, we've had to focus on analytic sensitivity, and again, just highlighting the differences between devices. So these are they're on different scales, which is why they're two different graphs, but they represent the lower limits of detection on the FlexMap for several of the bead regions and analytes that we're interested in. And you can see, again, we have some important differences that are present between the different analytes across the multiplex. And then again, the scale here is quite low, much lower than on the FlexMap. And what you can see is that we have different limits of detection across devices. So again, a challenge that has to be met as one tries to develop a commercial product. We've just now recently moved into human sample assay validation, and so I'll just show you a little bit of that data, starting with spike and recovery experiments. Here's the heat map of human serum detection across the 6-plex. And you can see, again, the concentrations vary widely. This should be concentration, I believe. And then we do see, for most of the analytes, a robust effect of the spike, and the percentages, the spike and recovery concentrations are set here. The blue is the neat sample, the orange is the spiked sample, and you can see the percent recovery is mapped here. So again, we still have some work to do. We're close on the ranges, but still some work to do in sort of optimizing this element and the effect of the matrix and the dilutional effects. So our next steps in timeline are, as I showed you right now, we're sort of moving the assay into human serum sample testing within Sage, within the Sage laboratory. In the early part of next year, we're hoping to parallel that work in the CLIA environment with our partner, and then begin scale up and hopefully product launch as a novel diagnostic for vascular brain health in the second or third quarters of next year. So I'll just conclude there and say that I'm trying to show you that in a limited amount of time that silent brain vascular injury is a common and progressive disease that increases the risk for both stroke and cognitive impairment and dementia. That chronic vascular risk factors are a key driver of that through inflammatory signaling, and this can be potentially therapeutically targeted if we can recognize it diagnostically. And then again, important to develop analytically reliable biomarkers as a precursor to targeted therapeutic development. And then I hope I convinced you that our inflammatory composite score is a robust multi-analyte measure, both of the chronic cerebrovascular injury that one can measure on MRI, as well as the precursor cerebral edema that can be present before that disease is manifest. You can learn more about the lab or the company via the QR codes, and again, most of the work I showed is that of Dr. Sadaf Pucci. We're grateful to be partnered with Cell and Biomedical, our CLIA lab partner, and I just acknowledge the folks who have been doing the work there. And of course, our corporate partners, Luminex and Biotechne. I'll stop there and happy to take questions.